Hi, everyone. Welcome to our live stream series we're calling Meet a Scientist. The Youth BIOlab at the Albertson Research Center in St. Boniface Hospital in Winnipeg, Manitoba is on Treaty 1 territory, the land of the Anishinaabe, Cree, Oji-Cree, Dakota, and Dene people, and the homeland of the Métis Nation. This week, I'm coming at you live from my home, and today we're going to get to meet Danah Alhattab, a master's student in a PhD student now in Dr. Mike Czubryt's molecular pathophysiology lab, which is part of our Institute of Cardiovascular Sciences. I'm also keeping an eye on our chat, so if you have any questions for Danah, you can join us there too. So I'd like to welcome to the show. This is Danah working in Mike Czubryt's molecular pathophysiology lab. I'll stop the share so that you can see a bit more of her talking with us too. So welcome to the show, Danah. Thanks for making some time for us today. So to start out, we often just like to ask our scientists from the research center things like, what brought you to science? Were you always interested in it growing up, or did you know you wanted to be a research scientist? How did you get to where you are today?
Hi, first, thank you for having me with this nice BioLab program. So I want to say that I've been always interested in being a scientist, and I was like, although I didn't have much experience in the research, in the techniques, but I was always interested in how scientists would find things and investigate things and like get drugs done. So like that's what I wanted to learn. And although I came from a different background, my bachelor degree was in dentistry. But like after working for some time, I thought like it would be really interesting to look for more challenging and solving some puzzles by looking into heart research in basic. Yeah.
So you have a degree in dentistry already?
Yes, I got the
And
degree.
did you practice in dentistry for a while before deciding like, oh, maybe I want to know a little bit more about what's going on behind these different problems that we're seeing?
So yes, I practiced dentistry for 4 years in Jordan. And then I
Okay.
decided to come and join the research center and like research in the heart. And where to go better than coming to the research center, St. Boniface Research Center. It's well known, so that's why I came here. Yes.
Yeah. So like if you're working as a dentist in Jordan, how did you find out about the interest of this research center or Mike's lab in particular, Dr. Schubert? Like, how does that connection work from, studying in a different field to saying, oh, actually, I want to switch gears and pursue benchtop science, and like that's the lab that I want to work in? How does that happen?
So, Dr. Schubert's lab is one of the well-known labs, and it got like a very high reputation with all the papers. And I really got interested in the heart research in basics and like understanding the mechanics of the heart and how it's going. I wasn't really aware of like Winnipeg in general or how living in Winnipeg would be that cold, but I felt like it would be interesting to discover new things and have like new challenges. And I didn't have experience in research, so I just why don't we try it? And it was really a nice experience. That's why I started my master's with Dr. Schubert, and now I'm completing my PhD with him as well.
Okay, so if you already have a dentistry degree, when you apply, so you applied to the U of M to work with Dr. Schubert, is that how it works? And you have some coursework and then some actual
research work
in the lab?
Yes.
Okay. Is there anything that, was unexpected or different than you pictured it would be, like from your ideas of, oh, I want to go into more of the science field, and then what is it actually like compared to what you expected?
Well, first, like changing from from one country to another, it was a little bit of a challenge for me and for my family. So it's a little bit, as I told you, it's more colder than being in Jordan, and also it's very it's a new language for us as well, like adaptation in the life style. And also like we thought, like when you read a paper or an article, we feel that, oh, okay, they did this and that, and you wouldn't value the amount of effort and amount of like lot of disappointments to reach to
Mm.
that point where you get the data that you would be sure that you want to represent in your paper. So this is one of
Mm.
the parts that it's really hard and challenging in being part of science.
Yeah, I think a lot of people do sometimes have the idea that scientific discovery is more like, this is what I think will happen, and then I'll do a test to prove myself right, and then I have that solution. But I've heard a lot of people say, some of the more important discoveries in benchtop science is what doesn't work, or like when you
Exactly.
think that you're going to get these results and then it doesn't work, and then you have to ask, well, why, and what's going on, or what are the alternate
Or
solutions?
like the troubleshooting, why did this go wrong, what's happening with this? And sometimes
Hmm.
you will get new discoveries with that, and you will be very happy. So it's different from one part to another. Yeah.
Yeah, so complicated. So you became interested in hearts as your focus before even coming here necessarily. That's how you found Dr. Schubert's lab. What have you been working on with him?
So our lab, we basically like we can call our lab as fibrosis lab. So it's a lab that we will look at the molecules and how they affect the fibrosis process. Well, fibrosis, from the word, it's like a lot of proteins and fibers that they interact and make like a form of a mesh that would be over the heart. And our lab has been working on like how this fibrosis could, like if there's injury in the heart or heart attack, and this like a kind of scar or a patch that would make the heart uh, not functioning well and not be able to pump in an efficient way for the
Hmm.
so we looked at a certain protein, which is Caraxes, in our lab, and how this protein would affect the heart in its ability to function like less like improper. And then if we remove this, or to make this key target protein removed, what would happen? Would that improve the function of the heart to make it pump properly? And we found that
Hmm.
yes, Caraxes helps in the buildup of the proteins that would form the mesh that will
Hmm.
impair the heart function. Yes.
Okay, so just as a review, I think there are a lot of words in there even that are a bit complicated. So to paint a picture of it, like your lab is looking at, the bad effects that happen with heart disease and specifically fibrosis. But you sent me a paper, and we've talked with Michael a bit before about high blood pressure being involved. So like when people hear heart disease, there are like all these different things that can happen to people. So high blood pressure, we know, is part of it, or you mentioned heart attacks. But the idea is, any injury to the heart changes those tissues. And
Yes.
so you had said, so you're studying fibrosis. So that's the reaction of the heart creating more fibers and getting stiff and tough. So
Yes.
when I was looking at some of the research, I Googled scleraxis just to make sure I had a clear understanding of it. And it seems to be in lots of different parts of the body. And like the cells that create the matrix, the gluey stuff, they're in different parts of the body too. So scleraxis isn't only something that's in the heart when the heart is suffering, but can you tell us what does it seem to be doing? Like, it's bad, right? Scleraxis is bad.
Well, it's not that bad. Well, let's say that during the progression of, for example, in the development, the scaraxis is there. There is like the
Like
ability of
from
the
an
scarax.
embryo,
Yes.
like when
So
babies are developing. Okay.
yes. So this was part of my master's degree project, which is the ability of the scaraxis to differentiate some cells, or make more cells from epithelium to mesenchymal. So these types of cells to make them more
Hmm.
specialized. Also, it has the function of changing the cells within the heart from fibroblast to myofibroblast. It activates the fibrosis fibrosis
Hmm.
process as well.
So
So
I've heard before, I just want to go back because that differentiation, that's fairly complicated. And if we have any students even in high school that are kind of learning about cell types and their jobs, their different tissue structures, like differentiation is the idea that some cells can still sort of shift what type they're becoming. Like if you go back to stem cells, those can differentiate into anything.
yes.
And you said, so I was under the impression that epithelial cells couldn't differentiate into mesoderm, but you're saying so some of those like skin or covering cells are triggered by scleraxis to become connective tissue forming cells?
Yes. So that's the point, and that's what happens during development, during certain diseases. For example, even during the they looked at this EMT, they call it EMT, epithelial to mesenchymal transition. So they looked at that. So it happens also in the cancer, in the metastasis. So what happens is
Hmm.
that there is some injury to the cell structure that triggers the scleraxis to turn on, and then the scleraxis would affect other proteins, and these proteins will be responsible for this process. So
Hmm.
yeah, so looking at the scleraxis, it's like we're looking at it from different aspects. So my master's project was different from like now my PhD project. My PhD project now, our lab has shown that scleraxis would help like scleraxis would affect the stiffness within the heart. And now what I want to look at is what scleraxis would affect the forming the stiffness within the vessels. Well, that's what we call like vascular fibrosis, you know. So
Okay.
yeah, and this like vascular fibrosis, it's usually happened in patients with high blood pressure. Like there is some dysfunction within the vessel wall that causes this to occur, and we want to see what if like we want to represent that in a way that if we have an animal model that has hypertension, and like we do that with mini pumps and with infusing with drug that causes this hypertension, and it's usually with people with hypertension, they have stiff vessels. So when
Hmm.
you have a stiff vessel, like the vessel become more thick, and there would be a mesh of proteins that would make the vessel thick and would impair the flow, the regular flow of the blood to end organs. And that's what happens in patients with hypertension. And we want to see what if we take out scleraxis at this point, would that help to improve the hypertension in patients with that? So this is what we're doing in an animal model for now.
Okay, so just to kind of reiterate some of that for people watching again, I'll share my screen again to just this little PowerPoint, just the idea that, you know, in a healthy artery, there's some smooth muscle in here. There are some connective tissues represented by these blue areas, and then there's enough room for the blood cells to get through in the lumen. The purple would be like endothelial cells. But so after chronic hypertension, so that means like high blood pressure over a long time, these walls of the arteries get thicker. And then you're saying also there are more cells within that muscular artery wall that create the gluey fibers.
Yes.
So if these would normally be really stretchy, what happens to that actual artery? So there's not enough as much room for blood vessel or blood to go through to get to the different organs. Are there other problems that arise from that thickened or stiff artery wall?
So this is the idea. We want to see exactly what scleraxis would affect the proteins that are forming the mesh. And this mesh could be in all different layers. And also in the smooth muscle layer, does it help in the proliferation, in the increasing the number of the smooth muscle cells within the artery? Would it make it harder for the vessel um, to like adapt the elasticity? So these are the things that we want to see. And if you can imagine like normal healthy artery, this is very nice image. Like if you look at the healthy artery, it's just like a balloon. So once it gets the blood, it's like inflates, and then if it want to spread the blood, it would contract, and it will spread the blood at a certain regulatory level to all organs. However, if you look at the hypertensive stiff vessel, you would think about it, it's like blocks of, or like it's a wall that it won't be able to respond to increase of the blood or decrease. For example, the body need for the blood and nutrients wouldn't be efficient as it should be, so that will make it more harder for the vessel to become more stiff. That's the image. Yeah.
Yeah, because I guess we often think, or like we imagine, the heart is the muscle that pumps the blood. So like we picture, the heart's doing all of the work, but in healthy arteries, those muscles along the tubes are actually helping push the blood forward. So not only are they stiffer and can't stretch, but they also aren't helping to push that blood forward. So there's probably going to be systemic effects. We had a question in our chat. Wondering, so how many people are affected by high blood pressure? Is it a big problem here, or is it a small problem?
So cardiac diseases in general are the main leading cause of death worldwide. And hypertension is one of like about 300 million people each year, like they get high blood pressure. And if you look at the high blood pressure, you would see that it's not like people take, for example, why we're looking for fibrosis and the stiffness, because usually people have controlled drugs to try to control the blood pressure, but after a while,
pressure lowering medication.
Yes, to lower the blood pressure, but after a while, after some time, these lowering blood pressure drugs won't be as effective as they started with, because there would be like dysfunction within the vessels themselves, and that would cause the irreversible damage that happens to the vessel, that not only lowering drugs would be able to help, but you need more drugs, or you need something adjacent to help in the regulation of the blood vessels and the heart as well.
Okay, so even the drugs that target high blood pressure, they might be lowering the blood pressure, but they're not fixing the artery walls.
Yes.
That's part of the problem. Okay. So are you looking at, so in your lab, you said you're looking at what happens if we take out or lower the scleraxis activity. Scleraxis is a protein?
Yes.
Okay, so but it causes all these changes that make the artery walls thicker. So if you take away the scleraxis, then maybe you think you might be able to reverse the damage in those artery walls?
Yes, that's what we're hoping to do. So, and we want to see, like, we're taking out scleraxis from each component of the vessel. So we're taking out the scleraxis from a model where you can have the adventitia, and we take the scleraxis and see if that brings the elasticity to the vessel, or we take it out from the smooth muscle cell and we see if that helps the elasticity. I haven't looked for the model for the endothelial yet, because that would be beyond my PhD. So yeah.
How long
we're looking at the function. PhD? So I'm in my third year now. So we have like this nice technique. It was really hard and it was very specific. It's called pressure myography. So I'm hoping that if students would come like after COVID to come and join us, they will see. So what happens is that we cannulate little little vessels from the mesenteries, and like we tie them, and we tie it from one side, and from the other side we add the pressure on a closed pressure system, and then you can see how the vessel, if it's like elastic enough, it would move. If you like block it from the calcium, and you remove the myogenic activity, it would move like a balloon. While if you have a stiff artery, you increase the pressure as much as you can, like for 140, and you will see only little difference in the movement, and that's why it will tell you exactly how the vessel is, like the components are stiff, that it's not even moving with the increase in the pressure. And I think it's a nice image. Like if you like look at it, it would be nice to illustrate. Yeah.
Yes, but I mean, the analogy that you gave before of it being like a balloon, that's pretty clear. You know, you could have like a really stiff, hard rubber balloon that's really hard to blow up, and that's your heart doing that work. Or you can have a nice flexible balloon that inflates easily, and that's probably easier on your heart and gets more blood out to your system. So I think that's a really good description. Are there any things, so you're using some molecular genetics or something to take out the scleraxis from your models? How does that translate into people? Is the future that maybe there will be a drug that people take when they have high blood pressure that can reduce the activity of scleraxis in their heart, or is it dietary, or is it what does that look like in people?
So we're starting with animal models, but we're hoping that this will give us like knowing these basics on how to affect the scleraxis will be able to help us to modulate this and be able to have this in the humans. Actually, our lab is working on like certain small molecules that could block the ability of the scleraxis to bind to other proteins in order to activate them and make it more able, increase the ability of the fibrosis to happen. So we're having this as a scleraxis could be a key target in our drug therapy, or in the drug design that we're planning to have to use in human models, probably 5-10 years from now. It depends on how things go.
That's interesting. I'm glad that you like brought that into the picture, the conversation, because like, you know, it's a group of people that are working together on like this area of the heart disease problem. So your project in the lab is trying to figure out, you know, like what are all of the actions that scleraxis does to say, you know, yes, we need to target this, or no, it's not actually going to be an important target. And then other people in your lab are working on what are the molecules that can affect the activity of scleraxis. How many different people, or like how many different projects would you say your lab is working on at the same time with different students?
So now currently we are like 3 PhD students, and we have 2 technicians, and each of us has our own project. We're looking at like wand healing, like not only the effect of scleraxis on the heart or in the vessels. We're looking at on the wands, like on the skin. We're looking at so we're looking at different aspects on how this scleraxis could work. We're looking at the mechanisms. So like you can say about like we have 5-6 projects going on in the lab. Yeah, it's a huge work, and it's a teamwork as well. So we help each other. We have like certain models that could we combine with each other on some help out, learning new techniques together on like we teach each other about the techniques, about we have like a group discussion meetings with our supervisor, and that's really helpful. We used to have like summer students as well, like they used to join our lab, and that I think that would be, if anyone is here, like anyone who's listening to me and like he's interested in getting into the field of research, it would be an amazing opportunity to get to work hand like side by side with scientists, with students, and maybe after getting to know your techniques, you will be able to get your hands on some your own project for like science fair or whatever. Like it is very nice, and maybe you would have your own paper with us. So that would be amazing opportunity to look for.
Yeah, so we get students coming through our lab sometimes, and we have directed a few students to, to email professors and tell them what they're interested in. And a few students that have come through us have actually worked in Mike's lab in the past as summer students. Is that also the path that you kind of had to take when you decided, oh, I want to study like cardiovascular health and maybe hypertension? Did you just email Dr. Schubert and say, like, I've looked up your research and this is what I'm interested in? Or
Yes.
how? And it's
I
just a real
applied.
person that said
I
fine, yes.
I applied for the for University of Manitoba, and I sent an email to Dr. Schubert as well. So it wasn't for me, like I didn't have the time to get like experience. I didn't have any experience in research, and it's not about the techniques that you want to learn. It's the whole thing. Even like I have learned new things by being in the lab with Dr. Schubert. Like for example, like giving presentations. I wasn't like an expert in that, and I'm learning that. So that would be an amazing opportunity. So looking for the I looked through the universities. I wanted to complete my studies in Canada, and I will look at for places where I can have like heart diseases, like investigation or research. And I wanted to get a degree for that, because my ultimate goal is to reach to a faculty position in academia where I can be able to teach students. Well, that's what I like, and also to run my own lab in the research, and that's what my ultimate goal for. So that's why I came here. Yeah.
So eventually you want to be like a new Mike Schubert, directing your own lab and instructing at university and giving all these presentations?
Yes.
That sounds great. So we've only got a few minutes left, but to wrap up, kind of, what are some of the things that you feel benefited you transitioning into science? Like you've already said, like if people can, you would suggest they try working as a student in labs. Like, what do you think has made that transition work for you into benchtop science?
Well, honestly, if you want to look for any student who would like to look for graduate studies, first they should have like the passion to be interested in the subject that you're working on, and to look for a good supervisor. A good leader would be able to push you forward, to be able to like write. Like for me, I wasn't like even the writing. I had the writing skills, but the scientific writing was a little bit challenging as well. And like being able to participate in meetings and be able to present your data, it's something that you need support. You need support from your supervisor, you need support from your colleagues and your lab members, and that's a teamwork that you need to be able to do work with, and that would really help me actually. And of course, the family support is the most important, and like my husband has been supporting me all the time for that. It's really challenging to be a grad student with family, but you keep your priorities. So yeah.
Awesome. Yeah, it's really, I think also people sometimes have this idea of science as just being an individual scientist working on their little thing, but it really is like an enormous team of people that are all kind of interconnected and sharing ideas like you suggested. So that's great. And Mike's lab, Dr. Schubert's lab, is very, very great. He's a great leader, I would say. I'm so curious. So that's definitely, like, you can see the passion in some of the people around the research center, that like that's part of their great leadership, is that they're so curious themselves.
Yeah.
Last question though, what do you think you'd be doing, if you didn't pursue science? Like what's your alternate life plan?
Well, I would stay as a dentist for I think, and I would probably complete my studies and become I love like teaching students, so that's what I like. So probably I would teach in dentistry rather than teaching in research and pathophysiology. So yeah,
Fair
that
enough.
would be, yeah.
Sometimes you really know your interests. So if it's body related or health related, then that's your true calling.
Yeah.
Well, I think that's probably all we have time for today. Thank you so much for sharing your ideas and your experiences with us, Danah, and good luck. When do you think you'll be done
Thank
your
you.
PhD? When do you become a professor?
Well, hopefully in less than 2 years, probably.
Okay.
Yeah.
Yeah. It's a long game, especially when you're perfecting techniques. But either way,
Yeah.
thank you for joining us for our Meet a
Thank
Scientist
you.
series. And for everyone watching, join us again next week. We have new scientists on every week. And we're not sure who next week is yet. We're hopefully getting somebody from the Institute of Cardiovascular Sciences, but you can keep up to date with us by subscribing to our YouTube channel for updates. You can also check us out at youthbiolab.ca and follow us on Youth BioLab on Twitter and Instagram. Thanks for watching and stay safe.
Thank you.
Bye.